PARP inhibitors are relatively new agents that are really we're just beginning to accrue clinical trials and to complete them. And the first part of the talk was looking at the evidence of the efficacy of PARP inhibitors in hereditary breast cancer, that is breast cancer that has been, that's related to BRCA1 or BRCA2. And the second part of the talk was the evidence for and against the efficacy of PARP inhibitors in sporadic triple-negative breast cancer. Triple-negative breast cancer being breast cancer that doesn't have the estrogen receptor, the progesterone receptor, or the HER2/neu growth factor receptor amplification. The reason why therapeutic development started in this field was two seminal observations that were published in Nature in 2005, one from Alan Ashworth's group, the other from Thomas Halliday's group. And they realized that by inhibiting a DNA repair process called base excision repair, this repair process takes care of the normal wear and tear on DNA that occurs in all of our cells at any point. So in an average 24 hours, there are about a few thousand occasions of guanine base oxidation, and base excision repair removes that damaged base and fixes the lesion perfectly. So these two groups realized that if you inhibited that process by inhibiting the PARP1 enzyme, what happens is the process goes halfway, it leaves a gap, and then when DNA is replicated as the cell divides, that gap is converted to a double-stranded break. And that double-stranded break is primarily taken care of by a process called homologous recombination. It turns out that homologous recombination depends on BRCA1 and BRCA2. So these two groups realized that this may offer a therapeutic opportunity in the sense that tumors in patients who have BRCA1 or BRCA2 mutations have lost BRCA1 and BRCA2 function. And so by inhibiting base excision repair, you accumulate double-stranded breaks. You don't have this process necessary for fixing those double-stranded breaks in the tumors. The nice part of this approach is that the non-tumor cells of these patients have plenty of activity because the fundamental event in tumorigenesis in these patients is the loss of the normal copy. These patients are born with one normal copy of BRCA1, one mutated copy of BRCA1. So their normal cells are protected from the effects of this therapy because they have homologous recombination. Because of the normal copy of BRCA1, the tumors have lost that. So in the presence of a PARP inhibitor, the tumors accumulate unacceptable amounts of DNA damage and the tumor cells die. So there is now good clinical evidence, at least with one PARP inhibitor, a PARP inhibitor that is now owned by AstraZeneca called olaparib, that there is good efficacy that was seen in a phase 2 trial that was run by Andrew Tutt and published a year ago, suggesting that, demonstrating significant activity in BRCA1 and BRCA2 related breast cancer. There has been a hypothesis that the so-called sporadic triple-negative breast cancers, those that arise in people who do not have an inborn error in BRCA1 or BRCA2, which is the majority of triple-negative breast cancer. There is this idea that a fair number of those tumors may have some defect in BRCA1 or the BRCA1 pathway. The reason for that is that there are a number of similarities between the characteristics of the sporadic triple-negative breast cancers and the BRCA1 related breast cancers. This has led to a great deal of enthusiasm to try to target the BRCA1 pathway in these tumors to see if we can kind of extend the results that we've gotten in hereditary breast cancer to the sporadic triple-negative breast cancers. It turns out that by some measures this has been somewhat successful. If you look at the activity of cisplatin, which is a conventional chemotherapeutic drug that targets cells that have lost homologous recombination, as well as other cells, there is activity of cisplatin in sporadic triple-negative breast cancer. However, it looks like there's not going to be a lot of patients who are going to respond who have sporadic triple-negative breast cancer to PARP inhibitors as monotherapy, that is PARP inhibitors given alone. There have been just a small number of patients treated so far, probably around 20 in total, but that's enough for us to know that there's not going to be a large signal. Whether or not there are going to be small subsets of patients who will benefit, I think we have to wait for larger trials. All that being said, when medical oncologists are faced with a situation where there's a tool that doesn't work so well, they tend to combine it with other drugs. And so there's now a fair amount of clinical development combining a PARP inhibitor with conventional chemotherapeutic agents. And the idea is that since, in general, broadly speaking, PARP inhibitors inhibit a branch of DNA repair, and they may then potentiate the effect of standard chemotherapeutic agents that damage DNA. And it's early at this point that we don't have the results of those sorts of trials yet, but there are a large number of them that are currently going. In non-hereditary, that is sporadic triple-negative breast cancer in patients who don't carry a BRCA1 or a BRCA2 mutation, it's unlikely that a large percentage of such patients are going to benefit from PARP inhibitor by itself as monotherapy. But it's way too early to say whether or not combinations of PARP inhibitor and conventional chemotherapy might have efficacy in that subset of patients.